So let's get this over here. And so it's going to be following with the work we did on microscopes in Lab 4. And it's going to be a homework assignment. And I have on here 11 different types of microscopes. And I'm going to ask you as your assignment to pick 5 of these and learn more about them. Save whatever website links you use as sources to make a bibliography. And for each of the 5, write a separate paragraph that describes again how this method works a little bit, and then an example in biology of when this method would be used. And so we're going to start with this, and we have to start with these 2 general types: electron microscopes and light microscopes. And we use a light microscope in our class called a bright field microscope. And we're going to talk about electron microscopes last because they're well known, out in the general public, but they are only used in very kind of isolated and specialized cases in biology. These are all examples of light microscopy, and so they're all used on a microscope that's similar to the microscopes we used in our course, but there are additional features. This first group of these have just a regular light microscope like we used, but they have some ways of filtering or altering the light path to create different visual effects. But again, it's a light microscope. Then these are all an example of a kind of microscopy called fluorescence microscopy. It's still a type of light microscope. It uses visible light that we can see with our eyes. But what is going on here is the sample gets stained with specific pigments called fluorochromes that can glow and produce light against this dark background. So we'll get to those. But importantly, these are all light microscope tools. These are all electron microscope tools. Electron microscopes are really expensive, and they're just not very common because they only have specialized uses. These microscopes can be all expensive. These start to get extremely expensive, $500,000 like that, and very fancy imaging. They'll typically collect a digital image, and that matches up really well, with a computer's ability to store that information and analyze that information. So let's get started just with a reminder of bright field that we talked about in our class. And so we start down here. No, let's start up here with your eye. And there's the ocular lens or eyepiece, and we had a binocular microscope, so 2 eyepieces. They almost always have a magnification of 10x. And then down here close to the specimen, we have the objective lens, and that varies on our scopes. I think we had 4x, 10x, 40x. And to get the total magnification, you multiply the 2 magnifications together. So we would have at low power 4 times 10, a total of 40x, 40 times magnification. And then we also learned that here's this, the specimen on the stage, and we learned that below the stage, under the specimen, there's another lens, a magnifying lens that's called the condenser. And the condenser is going to focus the light on the specimen. So the light actually comes up here kind of like this, and then goes through this condenser lens, and it focuses it on the specimen. The condenser lens can go up and down in the focusing process, and there's a little knob to raise and lower the condenser. And so that's bright field, and bright field is just wide open light coming up to your eye, and you see the entire field of view, and it looks like a bright light. Okay? And here's some examples of bright field. And these are samples that have been stained, and they've been stained with different dyes. And they're visible dyes, they're pigments. One good way to think about this is that in, let's say, the mid-1800s, chemists made some really strong progress in chemistry by identifying and starting to synthesize pigments that can be used as dyes in clothing. And some of those same dyes could also stain samples for microscopes. And so they improve the quality of seeing cells in the microscope. And they could, these dyes could stain both clothes and tissues, because the clothes at that time, they were all made by natural fibers, biological fibers like cotton and wool and so forth. And so it was staining biological material. And so, yep, some of them could also work well to stain samples for cells. And so that's an example of the bright field microscope, wide open view. So the next one we're going to talk about is a neat one because it's just the opposite of that bright field. And it's called dark field. And again, it's just a light microscope, but here's the difference. Here's the condenser lens, here's the specimen, here's the objective lens. So there's your specimen. And so in a dark field, what happens is under the condenser, a scope that can do dark field, there's a turret, and it's able to move different shapes into the light path. So we saw up here a term for a condenser turret, so it just turns around to move different objectives into the light path. Down here, some scopes have a condenser turret to move different shapes into the light path. And in dark field, a big circle is moved into the field of view, and it blocks almost all the light. And so it's a circle if you look down at it, and it's just a completely dark circle. And what happens is, it's shown here, is that blocks all the light in the middle, so that when the light gets focused on the specimen, none of the light comes up to your eye. And that's why it's called dark field. The field of view looks just black, like total blackness. And what happens is there is a sort of ring of light. So again, remember, this is a circle. If we look down at that filter, and there's kind of like a ring or donut of light that's coming up, but see how it's coming around the edges. And so if it comes up and it doesn't hit the specimen, it misses the objective lens. And this is a reason why the condenser needs to be focused up and down just right for these different kinds of light microscopy where there is a filter, is because we got to get this just right. Because what's going to happen is that if this light is affected by the specimen, the light could get bent, it's called refracted, and some of the light would come up into the field of view. And so what happens is you can see some features of a specimen, and they look like they're bright against a dark field. And that just gives a very different view. And one of the most important features of the view is that you can sometimes see things in dark field that are too thin to resolve to see as individual items in regular bright field. And so that's our first example of modifying this. And so here's just an image of a organism under dark field, and you can see how it's all dark except for some features of this. And some of these, like these little bristles here or these little bristles here, they're actually too thin to be seen with a regular bright field light microscope, even at really high magnification, because they're just below the resolution that a light microscope could do because of the limit of the wavelength of light. And here we can see some of these items that would just be impossible to view. And they're viewed here because we're not really seeing the object, we're seeing the refraction of the light. Okay, here's another image of dark field. Here's another image. These are sperm. Sperm tails are famously too thin to be seen really typically by a regular bright field light microscope. So that's dark field. Then the next one is phase contrast. And phase contrast sounds a little bit like dark field. And the purpose of phase contrast is to be able to visualize things that are not normally being easily visualized in bright field. And phase contrast also takes advantage of this idea of moving just a block in front of the source of light. And in this case, it's going to be another donut of light. And so here's kind of a complicated view of that. And so and here's an image of a phase contrast specimen. And so here is just the light source, and it comes up through here. And this is that block. And there's a disk like this right here that would be similar to the dark field disk, but it's not as big around. But if you look down at it, if you could see it through the microscope, it's a dark like this, dark ring like that. And then there's a ring of light. I could probably draw that better, folks. And then a thin ring of light comes through. And outside there also is this dark plate. And so we again get a donut of light. And what happens here is that donut of light is going to come up here, get focused on the specimen, and the specimen is here on the stage. And so some of this light is going to come up as like a ring of light through here. And then in the objective, inside the objective, behind the objective lens, there's a similar donut shape of dark light. And what we're doing is we're just making it more difficult for the light to make it up to your eye. And so similar to the dark field, we see some light differently because of its ability to get up to our eye. And what's happening here in part is that if the light gets refracted just a little bit, it won't make up to our eye. And so phase contrast is used primarily to increase the contrast. And unstained biological cells typically have low contrast. The cells are in a water environment, and water doesn't bend the light very much. And then the molecules in the cells have a refractive index that's pretty similar to water. So it just doesn't bend the light or reflect the light much. And so what does it here is if the light just gets bent a little bit, it won't make it up to our eyes. And if it doesn't make it up to our eyes, then that's going to look darker. And so that's how we increase contrast. Contrast is described as the difference between sort of a black and white image, that would be high contrast, and an image with like shades of gray and kind of gentle shades of gray, that would be lower contrast. And so you see shades of gray with most objects, biological objects, unstained under a microscope, because they just don't bend the light much. But if we make it very difficult for the light to go through the donut-shaped light and then the phase plate in the objective, then it's more likely that it will not make it up to our eyes, and the object will look darker and will have increased the contrast. It's called phase contrast because it's not just the light path, but also the phase is shifted. And that's just a little bit more complicated aspect of the optics that are happening here. And maybe if you have had a class in physics, you would choose to write about this and write a little bit about how it's not just the light path and the difficulty of the light getting up to your eye that's affected, but also the phase of the light itself. Okay, so here's an example of bright field for the stained sample, then dark field, and then in phase contrast. And in phase contrast, you can see things with more of a crisp, sort of darker, lighter image than here. And see, these little cilia are not visible very well either by dark field or bright field. And phase contrast is great. This is a stained sample. The pink is a stain. Phase contrast is typically used on cells that are unstained. And so see, there's an example of a low contrast specimen, a biological specimen looked at under bright field. Really can't hardly even see it. But then this phase contrast causes an increase in the contrast, and and you can see things in a more of a dramatic, darker, lighter way. Okay, so then the last one is also another story. We're going to just do regular light microscope, but we're going to use some special filters, and it's called DIC, or differential contrast microscopy. And what happens here is that there's a special, you'd probably call it a kind of a prism, that the light goes through before it reaches the condenser. And this prism splits the light into two parallel light lines. And I think of it a little bit as like how you have two eyes, and with your two eyes, you have binocular vision, and that's how we can see depth, is that we're really seeing things from two slightly different positions. And so the sample is going to get illuminated by light that's in slightly different positions. And so then those two light paths are going to go up through the objective lens. And then there's another prism that brings those two light paths back together. And so that's kind of the fundamental feature, is that there are these two prisms that first split the light paths and then bring them back together. And in doing so, you get a kind of three-dimensional effect. And for that, I have an image here, and it's a video. And this is a DIC image of a living, little tiny, kind of microscopic worm called a nematode. That's a popular research organism in biology labs. And you can see the cells. That's not stained, and they're visualized. You can also see how that's the nucleus. You can see how the nucleus looks kind of three-dimensional, and you can see some other features that have this kind of three-dimensional effect. And it's powerful because we don't need to kill the specimen to look at under DIC, and that was super valuable for scientists that study this nematode. Several scientists have won Nobel Prizes in this research organism for how they've learned things about genes in the nematode that are also similar to genes in humans, and learned about our health and disease from these simple model organisms. And one of the big advantages of the nematode is you could look at living nematodes and see the cells and watch them do their their functions in a living organism. So there's that DIC. So these are all a type of regular light microscope, and they just include a fancy prism or filter to alter the light path in order to and see things that you would otherwise not be able to see, or to see things with a better contrast, or to see things with kind of a different quality, to be able to really see things in more detail. Okay, so now we're going to talk about types of fluorescence microscopy. These are all fluorescence microscopy. The first one is just called fluorescence microscopy. Here's an image of it. And what's been done here is the sample has been stained with some tags, and the tags are like pigments, like a pigment, like a dye for your clothes that we talked about earlier. But these pigments are a little bit different, and they're called fluorochromes. And the pigments that are in our clothes, they absorb light and they hold on to it. And so, for example, like if there's a black car in the summer, if you put your hand on the car, you can almost burn your hand because the light has been absorbed by the black paint pigments, and it holds on to them. Another kind of light, another kind of behavior of light is to think about is you've seen like glow-in-the-dark toys, right? So you'll hold the toy up to bright light, turn off the light, and the toy will glow for a few minutes, okay? That's called phosphorescence. And the molecules in the toy can hold on to the light energy like the car with black paint, or other objects that have traditional pigments, but in phosphorescence, the pigments in the glow-in-the-dark toy, the kids can't hold on to the light permanently. So they hold some of it, the sample heats up a little bit, but then it's constantly releasing it. And so you can sort of activate the toy by shining light on it and then turn off the light. You see the effect. So fluorescence is different. Fluorescence is a lot like phosphorescence, except the pigments that absorb the light, so these fluorochromes, they can only hold on to the light for a split second, not long enough for you to turn off the lights and see that glow. But they're glowing like the glow-in-the-dark toy. They just glow really fast. And so the trick in fluorescent microscopy is that these pigments can absorb ultraviolet light, and UV light is invisible to our eyes. So the microscope has a light bulb that shines UV light on the sample. And That's called the excitation light, and we can't see it. So this sample is actually being like under a microscope with a really bright light on it, and humans can't see the bright light. So the pigments in the stains in this sample, in the way the specimen has been prepared, those can absorb that light and they can't hold on to it. So they're constantly letting go of the light, and they've held on to it for a short time, and so they'll release light that's at a lower energy, and a lower energy would be a longer wavelength. And so we start out with UV light shining on it, excitation light, and that can't be seen by us. It's a very short wavelength, but when the light is absorbed and then re-emitted as fluorescence by this stained sample, we can see those lights because they're lower energy and they're in the visible wavelength. So that's called the emitted light. We have the excitation light that's invisible to us, so we see darkness, and the emitted light is what we see. And scientists can pick different pigments to glow different colors. And you can see there's very interesting detail here. And so that's an example of that. And before we go on to these others, or actually I'll show this next figure. So we've already talked about these, just regular light microscope, and then we talked about a fluorescent microscope that has just a staining strategy. These don't require a staining strategy to function, and sometimes they're used in combination with staining, a pigmented stain like that. And then this requires that staining. But if we just look at these, they have something in common that this confocal microscope fixes. The rest of these are fancy microscopes that are types of fluorescence microscopes with extra features. And the first one is called a confocal. And a confocal does something to the visualization of the sample, of the fluorescent sample. And to understand that, we just have to look at this. So here's like a cell under a microscope. So this is, this is where the objective lens would be, and your eyes would be up here, and the light would be down here shining up, and that's the specimen. And you probably saw this effect in class or at other times when you use the microscope, that you can focus up and down through a specimen, and you see different parts of it. And so that's what's happening here. They lowered the focus through the specimen. And at first we just see the top part, then the middle, and the lower part. And the part that's in focus is actually got some thickness to it, and it's called the focal plane. And so we can look at these different focal planes. 3 are shown here. And then these are kind of pictures of those 3 focal planes. You're really seeing different parts of the specimen, same specimen, okay? So in the focal plane, those molecules are in focus. But think about this. The other parts of the cell are still present and visible, but they're out of focus. So like say, for example, when we're focused on this first top part, these other layers are still there. They're just blurry. And we can see the crisp focus on this, and it looks good. But this is called, these other layers are called out of focus information, and they decrease the quality of the image because they're there and they're just making the image blurry. They're not helping at all. And what a confocal does is, by using the fluorescent dyes, the confocal can not excite these layers, not cause these layers to glow with those fluorescent pigments, glow red or green like that, blue like that. They don't glow at all until the microscope focused on them. So we're just going to excite the molecules in this layer and get them to glow. And that removes the blurriness. There's no kind of glowing blurriness that's out of focus. And a confocal can collect data. Confocal microscope is a light microscope I use with additional kind of features on it. And one of them is that the focus is motorized. Another is that a computer is running the microscope. And we can control it through the computer. We can also just program, and we can say, just take a series of pictures of the specimen, focusing down through it, and we'll get 3 really nice or more really nice pictures. And then the computer has a program to stack those together. and make an actual three-dimensional object that the computer on this computer screen on the monitor can rotate and show you the different features of that object. And so a confocal microscope can make really high-quality images. And so that was really revolutionary. Another thing to mention is that light microscopes they can do magnification that's very good for biology. You know, they can easily magnify specimens and see the cells in the specimen and the nucleus and you know some of the organelles and stuff like that. So it's a good level of magnification. We just need to kind of continuously try to increase the quality of the image. So that's a confocal, and the key thing about a confocal is that it's going to remove that out of focus information, and it's going to give us a high quality image, and it can also stack it. And another thing is, once it's on a computer, the computer can actually quantify the intensity of the different lights. And so we can start to do quantitative experiments about, well, how much protein is here if you see that much glow, and how much protein is present, you know, in another cell. Okay, so confocal is great, and the next two examples are types of confocal microscopy that increase the quality of the confocal. So a two-photon or a multi-photon microscope is a confocal microscope, and what they're doing here is a photon is a unit or a bundle of light, and so, you know, if one photon hits the pigment will glow. So what's going on here is, instead of having one photon required to get the pigment in the specimen to glow, here's a two-photon microscope, and here's a three-photon microscope. And what's going on there is, instead of needing just one photon to glow, it requires two or three to hit and trigger this glow. And that's of value for a few different reasons. One is the multi-photon microscope, the photons are lower energy, and so they have a longer wavelength, and so they can penetrate deeper into tissue. So the tissue is going to absorb the light, and so one of the limitations of a confocal is that you can't see very far into the tissue, and we could look deeper with this. Then another is that the efficiency of light produced is higher in a multi-photon microscope, and so we can see with higher clarity the specimens. And there are a couple of other features that I won't mention here, but it's just a two-photon or multi-photon is just a way to increase the quality and your versatility of a confocal microscope. Then our next one is also the same idea. It's called a light sheet, and it's pretty interesting to see. It's a confocal microscope, but here, see how the light is off to the side. We're used to having the light here and shining up through the specimen. Here, simple idea, just have the light shine from the edge, and our eyes, or the computer that's detecting, the sample, is up there, and the light gets spread out, so we have a sheet of light. And what's going to happen here is that the sample is going to get rotated as it's being viewed, and so we can look at a three-dimensional sample and have it rotated around, and then the computer program can put all the images together and build another three-dimensional model. The reason for a light sheet is that you can look at bigger samples under a microscope. And so here's an example of a really tiny fish embryo, and with this light sheet microscopy, it's going to be a fluorescent type microscopy. So we're getting that fluorescent glow because we stain the specimens with fluorescent fluorochrome pigment, and you can just see this is going to be a bigger specimen, and we can see in great detail that. And scientists are interested in that, right? They don't want to just take a little slice; they want to see the whole organism, or the whole, like, embryonic brain. And so Light sheet is getting very common because scientists are having a lot of success growing in a dish what are called organoids. And so, for example, a scientist can grow a little tiny kidney organoid in a dish. So they can take stem cells and grow them in a dish and then convert those stem cells into an organoid. And the hope is that when, they're just tiny now, but as we learn more, we can understand the kidney or other organs like the brain and so forth better, and then just and then build bigger and bigger organs potentially to replace. Give yourself a kidney transplant that's actually your own kidney that we grew in a dish, for example. And so these organoids are very small, but that's still kind of big to try to collect that image under high magnification in a microscope. And so it's very helpful for light sheet to look at some of these bigger objects. Okay. Then the last one we're going to talk about is super resolution. And I mentioned up here at dark field that dark field allowed us to see some things were actually too thin to see with the limited resolution of a light microscope. And their super resolution is going to increase the resolution to see things in greater detail. And there are some competing strategies for super resolution microscopes, and I'm just going to mention this one here. And so what's going on here is the idea that we'll just say these are two of those pigments that can glow. And these pigments could be very close to each other. And so if you illuminate them with a regular light microscope, you would not see them as different because they're so close to each other, they just look like they're in the same place. But with a confocal, it's possible to actually excite this one and not excite that one, and then excite that one later. And so what the confocal is seeing, and the person's never seeing this because the confocal is collecting this as a digital image, is they'll say, well, I saw a glow here, and then I saw a glow over here. And they can just keep watching that, and that's what's shown here is multiple sort of dots of identification. And these two objects here that are too close together can be resolved because we're not seeing them all at once. We're seeing little dots over time and assembling that into a final image. These other versions are kind of complicated, but you might want to write about these as part of your assignment, okay? And so here's an example of a super resolution image. And so they're really high quality and can be very high magnification and look at great detail, like say inside the cell and inside the nucleus. Now we're just going to talk about these major types of electron microscopy for biology. There are some other forms of electron microscopes. Sometimes they're used in engineering, like to look at metals and things like that. But in biology, they're really two mostly common, but again, still quite rare types of electron microscopes. And one's called transmission electron microscope, and the other is scanning electron. And in transmission electron microscope, the electrons are going to be used instead of the light, and so the electrons are going to be used to illuminate the sample, and maybe the electrons will be bent, and you won't, see that light in your, or the computer won't see the light as it collects it, but and that'll increase the contrast, the staining. And so here's an example of a TEM, a transmission electron microscope, and they're rare because they're huge, they cost a lot of money, they need a special technician to run them, and then most importantly, the specimen has to be stained with some really surprisingly strong stains, typically metals. And because it takes a lot to bend an electron, so we talked about how the light could be bent or refracted. Well, it takes a lot more of an object like a metal to really bend an electron out of the light path. Here's an example of TEM, and TEM is used to look at things at very high magnification. That's its main thing. And this is at very high magnification. These are bacteriophage, so that's one right there. That's a bacteriophage. and they infect bacteria. So these are viruses that are extra super tiny, and you wouldn't even be able to see them. Look at this though, because this is the case here for this type of bacteriophage. It has a head, and that's where the DNA is that's going to get injected into the infected cell. Then here's the infected cell membrane, and this virus is going to land. And you can see how it looks like a little bit like a spaceship with legs. And these are real legs. That's not an artifact. They're very thin, and they're just made up of chains of protein. But they help orient the bacteriophage on the surface of the cell and land on it. And then the DNA is going to be injected through that stem into the target cell to infect it. So scientists, virologists who study these bacteriophage viruses, they are probably the most common reason that I know of for a scientist to want to use a TEM, transmission electron microscope. And these images are used to help identify the species of the virus that scientists are studying. And the last one we'll talk about is a scanning electron microscope. And a scanning electron microscope is not a big unit. They tend to be really small. And they they're used for a very different reason. And so a SEM, a scanning electron microscope, is used to look at low magnifications. So, we usually think of an electron microscope as high magnifications, but SEM is to look at low magnification. A really common thing is to look at the surfaces of organisms like insects. And that can help in a variety of ways to identify the species in detail or to look at the features of its body. And in SEM, the way that works is we actually have to spray, it's kind of fair to say you kind of have to spray paint the specimen with a gold or other metal, and that's going to block all the light, and we're going to look at light that's bouncing off that painted surface. So the transmission electron microscope lets light go through the specimen. Scanning electron microscope has been painted. The specimen's been painted, and so the light bounces off it, and we see that. Okay, so we're going to just do that. I'm going to stop right here and do a follow up on this, and thanks very much.